Hey everybody, it's Mr. Caudale. We're almost done with the semester, and specifically almost done with chapter 17. We have already started chapter 17, our lecture on it, and we got through kind of the basics and then transfer RNA, recharging of the transfer RNA. We just have to go through the nuts and bolts of how translation actually occurs here. I don't think I'm going to say very much about 17.5. Whoa, it keeps going. 17.5, it's all about mutations on the DNA, which then lead to changes in amino acid sequence, which leads to changes in protein structure and function. It's not to say that it's not important. I just chose what I wanted to talk about in 17. The reason why I say we're almost done is because once we're done with this, we'll go to inheritance patterns like, those Punnett squares where there's like a big B and a little b, all that stuff. And there's obviously a few more layers to that. And then we'll go into the newest genetic stuff. And that's it. That's it for the semester. And then next semester, we'll begin with evolution. And we'll be out of the cell forever. Some of you who are like art majors and history majors and psychology majors, never take another science class again. And you will never, ever go inside of a cell again. Some of you who are biology and biochemistry and physiology majors, it's just beginning, which I think is cool. Don't get me wrong. I went to college for 9 years, but there's something in this class for everyone, I think. So let's get moving. And I think the way we should get moving is you should press pause. And when you do that, copy this sequence down, and then let's figure out, let's imagine that this is a non-template DNA sequence read in a 5 to 3 direction. Sorry, don't copy what's on the screen, copy what's on the board, because this is what I'll fill out. Let's figure out the template strand, the mRNA, and then the amino acids that are associated with it. So go ahead and press pause if you'd like, and then I'll try to do it up here on the board also. Never made the same mistake I've made. Yep, that's right. It's 6:45 in the morning, so maybe that's my reasoning. Remember, somehow you start, it's how you finish. I even have a cheat sheet here. That's not really helpful. Now I'm looking at this codon chart. Actually, I have a codon chart for you. Here it is. Anyone see any problems with this? Yeah, don't be a wise guy. Just kidding. I'm sure there's. I would have gotten a C on the test. Let's do this. Let's pause again. Really take a minute or two with this and go over all what you know. It's always important to access pre-existing knowledge and then reinforce recently learned concepts, and that's going to help you layer here. This is a big picture that exists in the book. So I'm going to stop, go over what you know on the picture. Well, it is pretty cool when you look at this. You know a lot about this whole process of turning the information in DNA into actual protein that will enable the body and the cell to do any job that it needs to. We just need to figure out the last little bit down there, that ribosome that's in the middle center of the bottom. So let's get there.
In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site. There is also a third site called the exit or E site, where transfer RNAs are released. The initiating transfer RNA carrying formal methionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formal methionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of one codon, and the transfer RNA that carried the formal methionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of one codon. As the shift occurs, the two amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site.
That's wrong. The amino acids don't do that.
Continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
Okay, so here's what I'm getting at. So there used to be some debate. So this is what's neat about science or any other discipline, is that you think something and people are like, yes, this is my theory. And then it's proven wrong. And that's okay. That happens all the time in science. That's why it's so hard to create a law like Einstein's theory of evolution is now becoming a law. And the point is that people have theories, but theories are there to be proven wrong. Like the Earth is flat. That's what everyone thought. Or that the universe rotated around the Earth. And we know now that neither of those things are correct. And so those were theories. They were proven wrong, just like this. So there's two incorrect things in that movie that's about 10 years old. Number one, scientists now know that it's not the ribosome that moves along the mRNA. It's the mRNA that's pulled through the ribosome. I guess it's kind of like a perspective issue. Is the car moving down the road, or is the Earth rotating underneath the guy? I don't know. But the point is that now we know this is probably because most ribosomes are bound to endoplasmic reticulum, where they do this translation thing, and so they're bound. I mean, I guess they can move around, but let's imagine that they're kind of semi-bound, and then the RNA kind of gets pulled through them like a tape. That's not true for free ribosomes that are floating out in the cytoplasm. Number two, I said that what happens there, they used to think that the amino acid chain, they called it the P site, I think. We'll get to this over here in a second. The P site's the middle site on the ribosome because it's the peptidyl site, or where the peptide bond was being formed. And they showed that in the movie where the amino acid would shift over from the A site to the P site. and then you get this growing chain of amino acids that can then fold into a protein. Now we know that, in fact, the whole amino acid, the growing amino acid chain, it goes to the A site, and that's where the peptide bond is formed. And then the whole thing translocates essentially the naked tRNA then translocates everything to the left. And I'll show you what this looks like. But just understand that the amino acid chain doesn't grow in the P site, it grows in the A site. I'll show you that. But let's get to the ribosomes first. So here they are. Remember, ribosomes are made up of two things, rRNA and proteins that have come together. There's this thing called the nucleolus, which is this really darkly stained section of the nucleus in any eukaryotic cell, and that's where the cell stores rRNA until it needs to be used. Now there are a lot of ribosomes that work on the same mRNA at one time, and you'll read about this in the book. It's called polyribosome. All that, it's just a phenomenon where you have about 50,000 ribosomes per cell, and they're all translating mRNA at once. So what you'll see, don't imagine just one mRNA with one ribosome going down it. Imagine an mRNA with many ribosomes all going down it at once, because it's a pretty big moment when a gene has been transcribed and then is about to be translated into protein. And I think the cell wants to economize that moment and be as efficient as possible and make as much protein as possible, because those dicers are there cutting down the back end, the 3' end of that RNA. It's going to be degraded at some point. So that's what's happening there. So here's kind of what the small sum unit looks like, maybe from an electron microscope that they've kind of transferred into a cartoon here. Those are just amino acids. You've seen what proteins look like before. There's the large sum unit made up of proteins and rRNA. Well, there's 3 binding sites here, and I've written them up on this board. Why don't we go over to this board? So I always remember this as the Environmental Protection Agency, EPA. That's how I remember it. E stands for exit, and A is the first letter in the alphabet. So I'm going to tell you, this is where the first transfer RNA comes in. The A site's the acceptor site. It holds the tRNA that carries the next amino acid to be added to the chain. This is where so this is a codon right here. These 3 letters on this mRNA, which is read in a 5 to 3 direction. Remember, it's being pulled through the ribosome like a tape, and so whatever letter, this isn't right. So I knew I had them really tight here for some reason. So this is better. So whatever these 3 letters on the mRNA say, this is a codon here. That's in the A site, and then a specific tRNA is going to come in there and bring the next amino acid that's going to be added to the chain. You can see that the P site here is the site that holds the tRNA that carries the growing polypeptide chain. And what I said earlier was that that whole I don't know which way to turn here, the whole polypeptide chain of those 4 amino acids there is going to go over to the A site and create that peptide bond. Here's what a peptide bond looks like. We learned about this early on this semester. Remember, it's when a hydroxyl group and a hydrogen atom from 2 adjoining amino acids are bound, water is liberated from that, and you get this peptide. The E site is kind of the ejector site or exit site where that now naked or discharged tRNA goes, and then it gets ejected, and then the amino acid tRNA synthetase enzyme will grab onto it and recharge it so it can come back and do it again. Go ahead and turn to your neighbor, tell them what you know about the ribosome and how this all works so far, please. Okay, so just like in transcription, there's 3 stages of translation: initiation, elongation, and termination. So I would circle a few things here. If you have a handout that I've given you maybe, or this is just a picture in your book, I would circle GTP. So it doesn't use ATP, but it uses GTP to assemble the 2 subunits of RNA ribosomes together to do translation. Remember, they're looking for the 5' cap, or that G3P cap on the 5' end, and that's where they kind of get together. And then at that moment, they start pulling the mRNA through, and they look for a specific sequence, AUG, on the mRNA. And that's always the start codon. And when that start codon gets here, a certain tRNA comes in with a certain amino acid, and that's always methionine. You can see it in the picture as MET. And that's the signal that we're beginning the making of an amino acid chain that will turn into a protein. There's the translation initiation complex way over there on the right side of your figure. This says, then the small subunit moves along the mRNA until it reaches the start codon. I think it pulls the mRNA. During the elongation stage, amino acids are added one by one to the preceding amino acid. You can see the next guy coming in there at the top of the picture to the A site. You can see that GTP is being used there to then, I guess move the amino acid, the growing amino acid chain over to the A site. You can see that little red arrow. I would circle that. That's super important, because that's then where the peptide bond is being created to add that now 5th amino acid to that growing chain. Each addition involves proteins called elongation factors. It occurs in 3 steps: codon recognition. Codon has to be recognized here. Then we're going to have peptide bond formation, which is what we're showing there on that picture, and then translocation. And translocation is just where the whole complex is now going to be moved over. You can see that. You can see the growing polypeptide chain on the tRNA that was once in the A site, and now it's over in the P site. See that. Take a look at what's happening there on the far left side of this picture. That's translocation. It's going to take some GTP in order to make this happen. You can see then the once naked tRNA that was in the P site is now in the E site up there on the left side of your picture, and then it's going to be exited or ejected. Well, this happens, many, many hundreds of times until you get this long growing polypeptide chain, and then eventually there's a stop codon that comes here into the A site. This is, as you can see, either AUG, UAA, or UGA. And then there's no tRNA which is attached to an amino acid that will bind to those. But there are these things called release factors. I know that water plays a role here. This is probably just a protein that comes in, and basically it just blows the whole thing up. You can see it says termination occurs when a stop codon in the mRNA reaches a site, the A site of the ribosome. The A site accepts this protein called a release factor. You can see that there's, again, some GTP that's used. And the whole goal here is to make a free polypeptide that can then fold. And so we want this to happen. The polypeptide is then released. It's going to go, if it's in the ER, it will get into a vesicle, and it will go to the Golgi body, where it will have final protein processing, which means some amino acids will be taken off, some will be put on, it will be folded, it might get phosphate groups attached, other things may occur. And the point is that we want a functioning, mature protein, and that's where a lot of different things occur to make the proteins unique and able to do their jobs. But if it's a free ribosome and this is occurring out in the cytoplasm, the thing will just fold, and then it will just start doing its job, because there's a lot of enzymes and proteins that work out in the cytoplasm as well. This reaction releases the polypeptide, and the whole thing comes apart. See that there.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, It is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying the specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases called an anticodon that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
Good job, everyone. So here's the last thing I want you to do. This is super important. I want you to press pause, and I want you to spend 5 minutes, put a timer on your phone for 5 minutes, and I want you to go over every single thing from memory that you've learned about chapter 17, transcription and then translation. Go through it super slowly. Do it now, please. Well, if you were in class, we would show some sort of sign of affection to our friends, because this is not an easy segment, 12, 13, 16, 17. So it is worthy of a hug. I don't know, hug your neighbor, hug your teddy bear, punch the wall, whatever you need to do. I really like to have a test at this moment before we get into inheritance patterns and genetics. So maybe we'll have a test soon. I don't know how it's working with your class. Thanks. But stay tuned. I mean, I'm sure I told your class, but just at this moment in time, I don't know how it's working.